My name is Guo-Ping Shi from Brigham Women's Hospital. I just finished a talk at this International Academy of Cardiology meeting, annual meeting, I think it's 23rd years, held in Boston. My topic is about adipocytes regulates the abdominal aortic aneurysm via promote the binding of IL18 to its receptors. A few years ago, we reported that IL18, which is an inflammatory cytokine, has two receptors. One is the traditional receptor called IL18R. Another one is the NCC, we call it, which is sodium chloride co-transporter. It's ion channel proteins. It's very odd that the channel protein can also be a receptor for IL18. So when we use the mouse model to induce aneurysm, we found that both receptors are required for the disease, and which means IL18 in this disease model basically using two receptors to contribute to the disease development. Beyond that, we also found that in the human and the mouse aneurysm lesions, we found that the binding between IL18 to its receptors, both receptor NCC, which is a sodium chloride co-transporter, and the IL18R, both receptors, its binding to IL18 requires a involvement of the adipocytes. So we did a few experiments. One is, which I thought is very interesting, is that when we transplant fat tissues, which is adipose tissue, white adipose tissue from donor mice to the recipient mice, followed by inducing the disease, we found those transplants fat can promote the disease development. And a mechanistic study, we found that the fat tissue or fat cells, adipocytes, we use it both ways. For example, in vitro cultured adipocytes as well as the fat tissues from mice. We found that there's one molecule called leptin, which is the molecule that controls food consumptions and obesities, and it's well studied in the endocrine systems, endocrinology field. We found that the fat tissues or fat cells release leptins to contribute to the binding of IL18 to its receptors. And when the mice received the fat transplants, those mice develop bigger lesions, a large aneurysm size. And in contrast, if we use the same fat from OB mice, which does not have the molecules, we showed no activity. Those fat lost its activities. Therefore, we conclude that leptins from adipocytes are important to promote the binding to the receptors of IL18 to the receptors. And we also, in vitro study, we found that either the adipocytes or fat tissues, white adipose tissues, and in cell culture systems, we can also prove the same concept that those fat tissues or those fat cells can use leptin to promote IL18 binding to either one of the receptors by inducing the expression of their receptors, like IL18 receptor or NCCs. So we can see, increase of expression of the receptors of IL18 in cultured cells. So in contrast, if you use the OB fat, which do not have the leptins, those show no effect in vitro. So basically, in conclusion, in both in vivo and in vitro data, we show that the adipocytes use the leptin as a molecule to promote IL18 to binding to its receptors. And and we proved that in both in vitro and in vivo animal models. I think that's interesting to the field, and combine both cardiology and endocrinology together to present this work in this annual scientific session.